two men can make a baby with both of their DNA without a donor, without adoption. Just biology.
And two women can also make a baby. But here's the twist. They can only make daughters. Biologically. Forever. No exceptions.
Wait, that's a rule? A chromosome rule?
Hold on. If you can manufacture biological children on demand, that is not a medical procedure. That is a market. That is a very lucrative, very scalable market.
It is mildly alarming that your first response to reproductive biology is a revenue model.
But what if a gay male couple accidentally put two Y chromosomes into an embryo? What happens to that kid?
Does it become super male? Double the testosterone? Is that a person?
It's like if you copied a key twice with no lock.
There is no lock. The embryo dies, Bobby. It dies in the dish.
Why? What even is a Y chromosome?
I'm calling it YY-Gen, disrupting reproduction. Series A closes Friday.
Stop. Nobody is making a YY-Gen baby. Nobody is making a super male. And this is not a market. It is an active area of reproductive biology research with serious, unresolved scientific challenges. What we are actually talking about is this. How do you take a somatic cell, a skin cell, a blood cell, any ordinary cell in your body, and convert it into a functional egg or a functional sperm? And what does that mean specifically for same-sex couples whose genomes do not contain the chromosomal combination traditionally required to produce offspring? By the end of this episode, you will understand exactly why two women cannot have a son using their own DNA, why two men cannot simply use each other's cells without chromosome engineering, and why a YY embryo is one of the most doomed biological constructs imaginable. Let's go. Welcome back to the Krusty Lab. I'm Dr. Seeley. We have Bobby Sponge, Kelpie, Crabzilla, Squivin, and Patar.
And today we are talking about something that sounds like science fiction, but is apparently just science.
Regular science that I am personally trying to advance from my shed.
It is mildly concerning that that sentence was said without irony.
The topic is induced pluripotent stem cells, iPSCs, and their application to same-sex reproduction. This is real research, partially demonstrated in mice, actively being developed for humans, and raising some of the most complex questions in developmental biology.
If you want to understand why your chromosomes are basically making reproductive decisions without consulting you, subscribe. Because we are about to audit your genome, and it does not look good for the Y chromosome.
Let's establish the foundation. Every cell in your body, skin, liver, neuron, blood, contains an identical copy of your genome, but each cell type only expresses a subset of that genome, which is why a skin cell looks and behaves nothing like a neuron.
Right, same recipe, different dish.
Adequate analogy. In 2006, Shinya Yamanaka asked a different question. What if you could take a differentiated cell, say a skin fibroblast, and reverse that process? Force it back to a state of pluripotency, meaning a state where it can become any cell type again?
And the answer was 4 transcription factors. 4. That's it. OKD4, SOX2, KLF4, C-Mike. 4 proteins in your skin cell has an identity crisis.
4 proteins and you unmake a skin cell?
Essentially, yes. These 4 factors, now called the Yamanaka factors, are master regulators of gene expression. When overexpressed in a somatic cell, they dismantle the epigenetic landscape that defines that cell's identity and reactivate the transcriptional programs of embryonic pluripotency.
So it's a factory reset.
More like a factory reset that takes 2 to 3 weeks, involves extensive chromatin remodeling, and has a reprogramming efficiency of roughly 1 to 5%.
It is mildly poetic that the reset button only works on a small fraction of cells, and Yamanaka still got a Nobel Prize for it.
He absolutely deserved it. It's one of the most insane findings in the history of cell biology. You broke the assumption that differentiation was irreversible. You rewrote developmental biology on a Tuesday.
I once reset a rotor and it still didn't work.
Patar, that is actually weirdly.
Moving on, once you have iPSCs, you have a cell that can, in theory, differentiate into any cell in the human body, including germ cells, eggs, sperm.
Eggs and sperm from a skin cell.
In mice, yes. This has been demonstrated. The Hayashi lab at Osaka University and later at Kyushu University produced functional mouse eggs from iPSCs fertilized them, and obtained live offspring in 2016, and again with improved efficiency in 2022.
Mouse pups from skin cells, born alive, healthy, fertile. I framed the paper. It's in my shed next to the CRISPR kit.
How many mouse pups, and what's the per unit production cost?
It is mildly inevitable that you would try to monetize mouse reproduction.
Let me walk through the actual biological pathway because it is not trivial. You don't go from iPSC to egg directly. There's an intermediate cell type called the primordial germ cell, or PGC, which is the developmental precursor to all gametes. In development, PGCs arise in the early embryo and migrate to the gonads, where they differentiate into either eggs or sperm, depending on the gonadal environment.
So the body makes a pre-gamete first and then upgrades it?
Correct. In the lab, the process is somatic cell, reprogrammed to iPSC via Yamanaka factors, then induced to become a primordial germ cell-like cell, PGCLC, using a specific cocktail of signaling molecules including BMP4, BMP8B, and TNF-SF, thyrosin B.
And then you put those PGCLCs into a gonadal somatic cell environment, ovarian granulosa cells, or testicular Sertoli cells, depending on which gamete you want, and they mature.
Right. For eggs specifically, the PGCLCs are co-cultured with ovarian somatic cells. They enter meiosis, form oocyte-like structures, and can be matured in vitro to a metaphase to oocyte, which is the stage where they are competent for fertilization.
And this works?
In mice. In humans, we can get to the PGCLC stage and partway into meiosis. Full in vitro human gametogenesis, completing the full process to a fertilization-competent gamete outside the body, has not yet been achieved with consistent reliability.
So the product exists, but quality control is the issue. Classic supply chain problem.
It is mildly uncomfortable when reproductive biology sounds like a logistics brief.
It's like baking. Sometimes the bread just doesn't rise.
Pata, the bread is a human gamete, but fine. Yes, that's the situation.
Now, here is where the chromosomal arithmetic becomes unavoidable. Human sex is determined by the sex chromosome composition of the fertilized egg. XX is typically female, XY is typically male.
Right, I know this. I passed year 9 biology.
Excellent. In a same-sex female couple, both partners are XX, their somatic cells are XX, their iPSCs derived from those somatic cells are XX. When you differentiate those iPSCs into sperm-like cells, because you need sperm to fertilize an egg, those sperm are derived from XX cells.
And here's the crunch. Sperm are haploid. They only carry one set of chromosomes. Normally, male meiosis produces two types of sperm, X-bearing sperm and Y-bearing sperm, 50-50.
But in an XX individual, there is no Y chromosome to contribute. Every cell has 2 X chromosomes. Every sperm produced from those cells will carry 1 X chromosome. There is no Y.
So, X egg meets X sperm equals XX equals...
Female, every time, without exception. 2 biological women using their own DNA via iPSC-derived sperm can only produce XX offspring. Biologically, a son is not possible from that genome.
That is wild. They have daughters locked in, genetically locked in.
So the only way to get a son from 2 female parents would be to bring in third-party Y chromosome material, which breaks the fully biological to both parents promise. That's a product gap. I'm noting it.
It is mildly strange to call the absence of a Y chromosome a product gap.
Unless you engineer one. You could theoretically take one partner's X chromosome, strip it of all X-specific regulatory elements, and build a functional Y equivalent from scratch. But that
is decades away from human application, involves rebuilding a chromosome from scratch, and is not on the table for clinical discussion.
I said theoretically. I have a 3D printer.
My printer only does labels.
Now let's look at the opposite configuration. A same-sex male couple, both partners are XY. The challenge here is the inverse. Making sperm from male cells is straightforward in principle. XY iPSCs can be directed to make sperm. But to make an egg, you need a cell that can undergo oogenesis. And oogenesis, at least in mammals, has historically been associated with XX cells.
So the problem isn't making sperm, it's making eggs.
Exactly. In mice, the Hayashi lab demonstrated in 2023 that they could take male XY iPSCs and chemically induce loss of the Y chromosome, creating X0 cells, single X, no Y, and then selectively expand cells that had undergone spontaneous X chromosome duplication to become XX. Those XX cells were then used to produce functional eggs.
Let that sink in. They took a male mouse, made its cells forget they were from a male, tricked them into becoming eggs, made a baby. The baby is fine. The mouse doesn't know. Nobody told the mouse.
The mouse doesn't know.
That mouse should be paying royalties.
It is mildly surreal that we are discussing the legal rights of an uninformed mouse.
The technical approach involved two things. First, suppressing the Y chromosome during reprogramming using a drug called reversine, which causes chromosome segregation errors and can result in Y chromosome loss. Second, using fluorescence-activated cell sorting, FACS, to identify and select cells that had spontaneously duplicated their single X chromosome to become XX.
Okay, I need a translation. What is FACS?
Flow cytometry with a sorting function. You label cells with fluorescent markers, in this case markers for X chromosome dosage, run them through a laser, and the machine physically separates the XX cells from the X0 cells, based on their fluorescence signature.
So you're sorting cells like a laundry sorter?
Like sorting coins.
Functionally, yes. You are selecting for a specific chromosome composition from a population of cells with variable compositions, then expanding only those selected cells.
Wait, so the X chromosome selection step is a cell-level sorting problem. That's just data engineering with cells. I've pitched worse things to VCs.
It is mildly unsettling that that sentence was true.
Bobby, you asked earlier what happens if you accidentally put two Y chromosomes into an embryo. Let me explain why that is a one-way ticket to developmental failure.
I want to know, is it like super male?
No, the Y chromosome is, evolutionarily speaking, a degenerate X chromosome. It began as a standard sex chromosome roughly 180 million years ago and has lost approximately 90% of its original genes through a process called Muller's ratchet, the accumulation of deleterious mutations in a non-recombining chromosome.
The Y chromosome currently has about 55 protein-coding genes. The X chromosome has roughly 800. Those are not comparable structures.
Wait, the Y is tiny?
The human Y chromosome is approximately 57 megabases. The X chromosome is 155 megabases. And critically, the X carries hundreds of genes that are essential for basic cellular function, not just sex determination. Genes like MECP2, which is required for neuronal function, BTK, required for B-cell development, factor VIII and factor IX, required for blood clotting, ATRX, required for chromatin remodeling, hundreds more.
These genes have no backup copy on the Y. They're X-linked, homozygous in males, meaning males only have one copy, and if it's mutated, they get hemophilia, Duchenne muscular dystrophy, fragile X syndrome. The whole catalogue of X-linked diseases exists, because males only have one X.
A YY embryo would have 0 copies of all of those essential genes. There is no redundancy, no compensation, no SRY triggering male development, just a catastrophic absence of hundreds of required gene products. The embryo would arrest in early development, almost certainly before implantation.
So it's not super male, it's just nothing.
It is incompatible with life.
So YY-Gen is off the table.
It is mildly satisfying when market forces are overruled by chromosome biology.
I did try to model this once. In silico. The gene dosage problem is catastrophic from blastocyst stage. There is no fixing it without engineering in an entire synthetic X chromosome.
2 of the same key. Different locks.
Patar, that is actually kind of right?
The second key doesn't fit anything.
Mechanistically accurate, Patar, yes. There is one more layer to this that we haven't addressed, and it matters for both same-sex couple scenarios. Even if you solve the chromosome composition problem, even if you engineer XX cells from a male genome, you still face a fundamental epigenetic challenge: genomic imprinting.
Okay, what is imprinting?
Certain genes in mammals are expressed from only one parental chromosome, either exclusively from the maternally inherited copy or exclusively from the paternally inherited copy. This is called imprinting, and it is regulated by differential DNA methylation patterns that are established during gametogenesis, during the formation of eggs and sperm specifically.
The maternal imprint is set in the egg. The paternal imprint is set in the sperm. They are different patterns, and the embryo needs both. The maternal pattern and the paternal pattern to develop correctly.
So if you use 2 eggs from 2 women,
You get 2 maternal imprinting patterns. The paternal imprint is missing. Genes that should only be expressed from the paternal chromosome are silenced. Genes that should be silenced paternally are overexpressed from the maternal copy. This causes developmental abnormalities. The Prader-Willi and Angelman syndrome loci are classic examples of what goes wrong when imprinting is disrupted.
So there's an epigenetic signature that sperm carries that eggs don't replicate.
Correct. And this is one of the deepest challenges in iPSC-based gametogenesis. Even if you successfully differentiate male iPSCs into eggs or female iPSCs into sperm, you need to ensure that the correct imprinting patterns have been established. The machinery for resetting and reestablishing imprints is complex, tightly regulated, and difficult to replicate fully in vitro.
In the mouse experiments, some offspring had abnormalities consistent with imprinting defects. The efficiency is improving, but it is not solved, not even close.
It is mildly relevant that the hardest part of making a gamete is not making the gamete, it is making the gamete correctly.
Like seasoning, you can't add it at the end.
Pitar, that is exactly right. The imprinting is the seasoning. It has to go in during the process.
I genuinely cannot tell if Pitar is a genius or has excellent cooking timing.
To ground all of this in current reality, in humans, in vitro gametogenesis from iPSCs is not yet a clinical procedure. We can reprogram human somatic cells to iPSCs efficiently. We can differentiate those iPSCs to human PGCLCs with reasonable efficiency. We can initiate meiosis in those PGCLCs. What we cannot yet do reliably is complete meiosis in vitro, produce human eggs or sperm with the correct imprinting and epigenetic state, and achieve fertilization-competent gametes outside the body.
So it works in mice but not humans yet.
In mice, live offspring have been produced. In humans, we are at the stage of inducing early germ cell specification. Groups at Kyoto University under Miteneri Saitu at Cambridge and at the Weizmann Institute are actively working on this. The trajectory is clear. The timeline is not.
5 to 15 years for first human iPSC-derived embryo in a clinical context is the estimate I see thrown around. I think that's conservative. The mouse-to-human translation for reproductive biology has historically been harder than expected.
But the commercial structure is already emerging. Clinics are positioning. I'm told there are at least 3 biotech startups in this space already.
It is mildly uncomfortable that the race to commercialize the technology has outpaced the completion of the science.
People always put the cart first. So, you take any cell, you erase what it was, you rebuild it as a germ cell, egg or sperm. The sex of the baby depends on which chromosomes go in. If you only have X chromosomes, you only get X outcomes. If you have an XY cell and want an egg, you have to remove the Y and duplicate the X first. A YY cell fails because the X carries genes the Y doesn't know exist. The imprinting has to happen during the gamete formation, or the embryo reads the wrong instructions. That's all of it.
Pitar, that was perfect. You understood everything. You understood all of it.
That was genuinely a complete and accurate summary. I have nothing to correct.
She never doesn't get it. She just waits until we're done talking.
It is mildly efficient.
I want to hire Pitar.
I don't work Fridays.
Okay? Final exam. You have been sitting there nodding or staring at the ceiling for 20-something minutes, and now it is time to prove it.
I'm adding a bonus question that doesn't have an answer yet, just to keep you humble.
Pause the video, answer the 3 questions, then press play. Question 1. What are the 4 Yamanaka factors used to reprogram a somatic cell into an iPSC? Name at least 3 of the 4. Question 2. Why can 2 biological women using iPSC-derived sperm only produce female offspring? What is the chromosomal reason? Question 3. Why is a YY embryo non-viable? It's not about sex determination. It's about something more fundamental.
Well, wait, take your time. No shame in pausing twice.
Take all the time you need. The mouse already knows the answers.
And we're back. Question 1. The 4 Yamanaka factors are OXT4, sometimes written OXT3/4, SOX2, KLF4, and CMYC. An alternative reprogramming cocktail uses NANOG and Lin28 instead of KLF4 and CMYC. Full credit if you named any 3 of the 4 core factors.
I got 3. I forgot C-Mike. I always forget C-Mike.
CMYC is also a proto-oncogene, by the way. Overexpress it for too long during reprogramming, and congratulations, you may have also introduced cancer risk. The field has mostly moved to C-mic-free reprogramming protocols.
So one of the 4 reprogramming factors is also a cancer gene. That's a liability disclosure I would want in the prospectus.
It is mildly on brand for biology that the reset button and the cancer switch are the same button.
Question 2. 2 biological women are both XX. Every somatic cell in their bodies is XX. iPSCs derived from those cells are XX. Sperm derived from XX cells undergo meiosis from an XX genome, producing only X-bearing sperm. There is no Y chromosome present to contribute. X sperm fertilizes X egg. Result is always XX, always female. There is no mechanism by which a Y chromosome appears from a purely XX genetic background without introducing external genetic material.
That's the part that blew my mind. It's not a preference or a probability, it's arithmetic.
Pure chromosome math. No exceptions.
Unless you CRISPR in a Y chromosome from somewhere, in which case it's no longer fully from both mothers.
Correct. The fully biologically from both parents condition breaks at that point. Question 3. The Y chromosome contains approximately 55 protein-coding genes. The X chromosome contains approximately 800. Many X-linked genes encode proteins essential for basic cellular function. MECP2 for neuronal survival, BTK for immune development, clotting factors, chromatin regulators, metabolic enzymes. None of these have functional equivalents on the Y chromosome. A YY embryo would carry 0 copies of all those essential X-linked genes. The result is catastrophic gene dosage failure beginning in very early development. The embryo does not develop. It is not about sex. It is about the hundreds of housekeeping functions the X carries that the Y has no knowledge of.
I said it dies. I got the right answer. I didn't know why, but I knew it dies.
Intuition counts for something in biology.
So the Y chromosome is basically a liability holding company with 55 assets and no coverage for anything the X does. That is actually how some of our portfolio companies are structured.
It is mildly predictable that the least informationally dense chromosome reminds you of your investments.